thank you also to the organizers for inviting me here. It's been a very interesting experience and a very diverse workshop that I really enjoy. So I would like to talk about something that we've been working on for a long time. Is actually related to the previous speaker, but I'm not going to explain the connection now. So the challenge of trying to infer under how much purifying selection variants are across the genome. So trying to break these silos that we've also seen here of drilling down from what we think is the most severe to less and less severe, and trying to capture that across a wide range. And so this is something that started quite a while ago when I was a postdoc with Jay Shenduri, and then continued in my own lab since 2017. And yeah, so the question that we're trying to address here is if we sequence a genome and we recover a number of variants from this genome, how do we prioritize those that might be disease causal, that might have a molecular effect? And these might be large numbers of variants that we have to sift through. And like I just said, this is, our goal was really to break up these filtering hierarchies that go, let's say, from nonsense to canonical splice, to missense splice region, and then slowly into the regulatory regions. And for each of those, have a bunch of individual annotations that they use, and trying to prioritize with these additional annotations. Because I mean, these annotations are pretty diverse. Everyone is using a slightly different one. Everyone is using a slightly different threshold on those, and it is just very, very complicated to navigate. So we're talking data integration essentially. So how could we integrate these different annotations on a genome-wide level? Well, if we had a training data set of pathogenic versus benign, and if this training data set was nicely distributed across the genome, there would be a very intuitive way of doing that. And fortunately, we don't have that. Right? The variants that we know, the pathogenic and the benign variants that we already know, they're not evenly distributed throughout the genome. They're highly clustered. They're clustered because of these hierarchies that I've shown you, but they're also clustered because of the way that we're studying genes and for how long we've been known a certain gene, how well a gene is conserved, whether it's present in a model organism or not. This all impacts the number of variants that we find in our clinical databases. So just because of that, they're not very well suited to be used. The number of variants in these databases is growing, so don't get me wrong. So there's there might be potential for improvement. However, so this is looking at Clin Var, and Clin Var still lacks exon distal variants. So this is a representation of variant categories, the ones that we're trying to abolish, across the genome. And this is Clin Var. So in Clin Var, you find a lot of variants that are obviously very close to exons, a lot of nonsense variants, and a lot of missense variants. And this is, there's some variation between the clinical labels or the variant significance labels that are in Clin Var, but this is probably even more indication of an ascertainment bias in this database. And if you might wonder whether this has changed at all over the last years, it hasn't really. So the representation of non-coding variants, even over just the last few years, hasn't improved in Clin Var. Okay, So what we did back in, I started 2012, then published 2013, is we decided to use these human lineage-specific differences that we partially just discussed in a different context. So variants that since the last common ancestor with chimpanzees, so the ancestral genome sequence, if we compare to the human reference, positions that have changed. There are about 15 million of these. And you exclude variants that you see, let's say, in 1,000 genomes at a time. So you get a nice set of variants that have that are fixed, derived in the human reference or in the human population if you use 1,000 genomes additionally, and for which you can assume that you've seen many generations of purifying selection. So some of them might be functional, but we would assume a very small minority, and the vast majority is actually fixed by drift and has seen a lot of generations with purifying selection. And now these variants are actually spread around nicely throughout the genome, and we can contrast them with variants that we randomly draw. So essentially we are drawing de novo variants with the same substitution patterns and approximately similar variant density in large blocks of the genome. And you can imagine the result to look like that. So in the human-derived alleles, the severe effects are underrepresented, and then you have these de novo variants that you draw and that we consider proxy deleterious. And you can now use those two as the two labels in your training data set, and you can use those to combine various annotations that you have. And so, like I said, we've done that a while ago, and it is actually, we think, very useful because you now get, let's say for a nonsense variant, you now have a score, right, that ranges from probably no effect, like olfactory receptor, to high score neurodevelopmental gene. And everything is on the same scale. You, in principle, don't need to consider the variant effect category anymore. It's part of the model. You're getting a continuous score with which you can rank your variants. So it has been popular, people have been using it, have been citing it, and we have tried to maintain it over the years. So we started supporting the new reference genome, put it on a new website, made tools, made offline scoring tools available. More recently, we've been trying to address specific limitations. So we've been looking into whether, for example, the splice annotations could be improved that we're integrating. And so we've been using experimentally available data to benchmark a number of tools that were developed for splice variants, especially also deep learning models, so convolutional neural nets that have been used. And we found that these are specifically very useful. I mean, it's very similar to what Tony has just talked about with 3D genome architecture. We get a sequence-based model where we can put both alleles in, and we can calculate a difference, a delta value for the potential change to the genome. And we can integrate these delta values in our model. And there are other specialized models becoming available for UTRs and for regulatory sequences, which I will briefly address too. So we can then see that just integrating these additional annotations improves our model performance in these specific categories. You can see here for two different versions. So here we had included just more static annotations about splice variants from DB-NSSV, and then we've been including the sequence-based models in this version here, and we get another boost in the performance for specifically these sites, while other variant categories are not impacted by that. So if we find a better annotation, we can put it into our model and we can integrate it into our variant ranking. So in principle, you could just continue in doing that for all molecular functions or molecular predictors that you imagine to work on a genome-wide scale that are sufficiently unbiased to be integrated. And yeah, it would keep us busy. But there are also other things methodologically that you can do. We've, for example, looked into alternative definitions of this training data set. So you can, for example, also use actual singleton variance versus as a proxy for this de novo class versus standing variation, and you get a similar model out of that. Yeah. So I at least wanted to mention, so what we've been looking into over the last years, also these regulatory sequences, which we also heard are very important, and it's really not a new thing. We know that since the 70s that these are probably underlying a lot of our evolutionary changes. It's a diverse class, and we're sometimes not very careful in defining what we're talking about. So I'm specifically now talking about not the bigger class of non-coding sequences, but regulatory sequences, so promoters, enhancers. And we've been studying them with massively parallel reporter assays. So where you it's similar to, in a way, a luciferase assay, where you have some reporter gene and you're putting the element of interest with a minimal promoter in this reporter gene, and you're reading out the activity of this element or the change of activity depending on which element you put in there. And so a few years ago, we've done that for a number of elements, and we've done in silico, no, sorry, we've done saturation mutagenesis, so not the in silico saturation mutagenesis that we just heard for these computational models, but actually creating all potential variants along these sequences and reading them out. And we thought this was a very interesting exercise because we were hoping that it would also tell us about useful predictors of regulatory function. And we realized that there's not so much useful out there at this point. So one limitation, so a lot of these tools are prioritizing species conservation pretty highly, but species conservation is not necessarily the best predictor. And a lot of methods, they're good in inferring repressive effects from destroyed motifs, but on the other hand, they're not really good in seeing the creation of new motifs. So I'm just putting two very brief examples here. So the one is where conservation is really not very informative. So here, up here, you're seeing the variant effects that we read out, and below you see some conservation tracks, and you see some functional predictors. Yeah, and then you see annotated motifs, and you see this is not very well aligned. So neither the available motif data is well aligned with these blocks where we see the largest effects, nor do we see a good correlation with the conservation scores. And it highly depends on the element. So this is another element where things seem to be much better predicted by conservation, but again, motifs are really poorly annotated across these regions. So there's a lot to be done, and so this is maybe a bit of a summary here about the power that you have depending on what you're looking at. So this one is the power that you have in missense variants for separating pathogenic versus benign variants, versus trying to predict regulatory variant effects. So there's still a lot to be done. And what the field has been looking at over the last probably already 7 years by now. Yeah, more. There is the convolutional neural networks. So you trying to use sequence and deep learning models to infer function, and we are seeing some attempts, and I think this needs to continue, where one integrates additional information into that, or like histone marks, or increases the context. So there's a lot that can be done. And so actually these sequence-based predictors, they are currently performing the best in these comparisons with the reporter assays. But again, here there seems to be a larger challenge in developing good models that also work across cell types, or that are profiting from training data in many cell types. Right now, we are seeing more of a mean expression prediction effect if you're trying to go into integrating data from many cell types and profiting from the data that's actually there. Okay, so then I would like to switch over from these rather short variants. So CADD can actually predict or score variants that are single nucleotide variants, that are short indel changes, that are multi-allelic substitutions. But obviously there's a whole class of much larger variants out there, structural variants, and I probably don't need to give an introduction into those, but I think the fun fact that maybe not everyone is yet aware is that if you just look by a number of bases affected, they are the class that describes the most differences between two individuals. So even though and this depends on the study, right? The first study suggested that we have about 10,000 structural variants. More recent studies are up to 25,000 structural variants per individual. So the number is much lower than the millions of short variants that we were just talking about, but the impacted sequence might be much bigger. And so again, the challenge here that there's not really a good training data set. So the Clin Var data set especially is very small and very, very biased. So we're trying again to move to a similar system here, where we use the human-derived and the chimpanzee-derived variants this time to have a proxy benign data set, and we match that with randomly drawn regions of the same size from the genome. So here, just to show you that again, that the Clin Var data set is really problematic. In gray, I'm showing you structural variants from Nomad SV, and then in red and orange, you see the Clin Var variants. So they are much longer than the typical variants that we observe. They're really, really close to coding genes, and they're enriched for haploinsufficiency variants or haploinsufficiency genes. So that's why we're trying to get away, right, from such variant sets and use this other data set for training. So it's a very similar system otherwise to CADD, but a major difference now is you need to summarize all these annotations across a wider span, and you need to make some design decisions about what you do there. So you have a bunch of annotations that you can obviously use, but then you need to ask, how do I incorporate them into my model? So you can incorporate them, some of them as distance features. So how far are we away from a TED boundary? How far away are we from the next coding exon? But you can also then incorporate some summary statistics, like what proportion of bases have a CADD score of, I don't know, above 30? Are there GERB highly conserved regions in this window that got deleted? It's actually a big challenge for inserted sequences, and we are not solving that very well right now. For insertions, we're currently just looking at the immediate genomic context, while what we probably should be doing is looking more into predictive models that use the sequence. Okay, so one featureizes those and then puts them into the model. There are not too many tools out there yet, and we've also been having problems comparing to the tools that are available because they are sometimes not easy to run or cannot even be run on a new variant set. But so the tool seems to generally work. Again, we try to pay some attention to also make it accessible, to have a website where people can upload their variants, have a tool that they can download, and allowing them to use that. You can then obviously apply such a tool, and even just on the Nomad SV data set, you can pull up interesting events that might be potentially interesting, also from a clinical perspective for these individuals. But so yeah, so it works as it's supposed to as a variant prioritization tool. Yeah, so that then also covers the second part. So we've developed a tool for the present human reference genome. We support older assemblies by coordinate liftover. We currently don't support inversions and translocations just because we don't have any training data for it. And in the future, we would really love to go into including the inserted sequences into the model. Okay, that's where I'd like to thank you, and I'm open for your questions.
Trying to integrate fronting,
Say the database names again?
fronting and also other databases that hold additional information than Clin Var and sometimes variants.
So we're not using Clin Var at all, right, for training. We just use it as a validation set at the end. So we're not training on it. So in that sense, it doesn't. So Vaso is the database that you're referring to. Yeah, no, we could look into that, but we're really just using it as a benchmark at the end of the day because everyone's comparing to that. But it's not part of our training data set to start with. Yes.
In the last version, you said it's not cancer structural variants, like next one. Fully ascertain them, or do you mean germline predisposition to cancer, or actual like tumor derived?
So these were sets structural variants identified in patients with cancer that we tried to score. It is deleteriousness is not the ideal measure for somatic variation. I guess that's where your question is also heading towards. Yeah, it works to some extent. It's similar for CADD with short variants. You can apply it for cancer. It is not ideal. The assumptions are not really met.
And then the second question, I think structural variants are fascinating, and I was wondering, do you think that you have estimates of how well you ascertain them with mapping to the genome to ascertain that?
So luckily, we're not the people that need to map their outer points correctly, right? People just have to report the variance to us. But yes, there's quite, depending on the method, there's a good amount of uncertainty about the ends of these variants, and we have made an arbitrary decision here of using 100 base pairs context. If the variance is even higher than that, we obviously have a problem. The good thing is that, I mean, the methods are also getting better, right? With long-read sequencing, we're getting much more precise breakpoints, and more people are actually willing to identify the structural variants of that. But yeah, a very valid point, especially if you look at old technologies, right? For identifying them, the uncertainty might be in the tens of kilobases. Yeah.
So you started out saying Clin Var is a bad set as deleterious variants, correct?
It is it has a very strong ascertainment bias.
Correct. As
So
the
you have to be
negative negative points, like deleterious. When you need training, you need positive, negative, right?
Yes.
So you're using Clin Var. You're saying Clin Var is bad as negative.
It's also bad as a positive. So if it used the benign variance from Clin Var, it also has problems.
Oh, okay. So instead, CADD is using the variants that are in human or chimp or your SB as positive, as benign.
That changed, right? That are fixed in these species. So let's say there's an insertion into the chimpanzee genome.
Okay.
That was not in the common ancestor of human chimpanzee.
Yeah.
Then it's part of our training data set. Or there's a deletion as benign, yes. Yeah. Because it has fixed within the last 6 million years and
told us yet. So probably
yeah, hasn't killed us, hasn't killed the chimp. Yes.
And then the rest of the genome, randomly chosen ones that are not part of the benign set are actually your deleterious.
Yeah, they're just randomly chosen, same length. Okay. Yeah. So for the structural variants, it was really just random. For CADD, for short variants, it is same substitution patterns, and also considering variant density across the genome.
Okay.
And we've seen that. So you can replace the original CADD training data set by using singletons, let's say from TopMat or NOMAD, and contrasting that with let's say variants above 1% allele frequency works as well. You can do that. So we've done that, and you get about a similar sized training data set.
It's wonderful. Maybe we continue.